Today we'll be talking about ticks and vector-borne diseases. So we'll be moving up in scale from the micro viruses to the macro ticks. So of course, we live in a world of zoonotic spillover and of the jumping of parasites and pathogens into human populations. And although viruses, which are probably our greatest historical nemesis and based on the present pandemic, still a major threat to us, despite the fact that they take up a huge amount of attention and interest and are particularly dangerous, another group of parasites has nonetheless not left us, and it's still of great importance, and these are the ticks and other vectors of disease. So in a world of zoonotic spillover, certainly here's two questions that I think are very important for answering, or we need to answer these questions. One, we need to work out very fast, cheap, and accurate methods for parasite identification and diagnostics. And we need to start forecasting the pathways of vector-borne disease spillover so that we can potentially mitigate those or control those so that we prevent spillover in the first place, or we can monitor it enough that when it does happen, we catch it very, very quickly. So first, let's talk about why ticks are such excellent vectors. Really, they are the dirty syringes of the natural world. So if we take hard ticks, for instance, hard ticks have three different life stages. They have a larva, a nymph, and an adult. And between each stage, they leave the host to shed their skin, which means that as they go through their life cycle, they will typically feed on three different host animals, sometimes of different species. So they're the ultimate vector for bridging pathogens between different species and between different individuals. They really are dirty needles of the ecosystem. But of course, we think a lot about ticks as vectors of disease, and of course, this is a microbiology thing, so we're interested in the pathogens. But of course, ticks are very important parasites which damage human health in their own right. So down in the bottom in the teal color, you can see that ticks certainly spread tick-borne infections, but they have other impacts on us. They have things like tick bite dermatitis, which is particularly nasty, tick bite anaphylaxis, which in some cases can be fatal, tick paralysis, which also often leads to fatalities in both humans and animals. The recently recognized tick bite-induced mammalian meat allergy, which is radically shaping the diets of sufferers around the world, and we've found it on every inhabited continent. There are, we know that it's been caused by numerous different tick genera and species, so it's not a small issue. And then we have more minor things like odoacariasis, which is the invasion of ticks, say, in the ear canal, which can cause dizziness and things like that, pain, whatever. So when I first got to Singapore, there was basically nothing known about ticks. The first species was described from Singapore. The first tick in Singapore was described in 1909. There was an ecology study in 1971. And then there was basically silence. So the 20th century has been not very productive for people trying to understand ticks or tick-borne diseases. So I arrived here and I was faced with this pantheon of different interesting tick species. We didn't know who they were, we didn't know who they were feeding on, who they were biting, and we didn't know anything about the pathogens that resided within them. So very fertile ground. And it also means that Singapore is a great case study in how other regions in the Asia Pacific can basically go in and spend a few years and understand their entire tick fauna to some extent. And I'm sort of working with some collaborators, but we're a small team. And so this is basically what I've done towards tick diagnostics in 4 years. And we basically solved the whole problem here. So if we can do it here, of course, Singapore's a smaller country, but we have quite a rich tick fauna. So these same methods can basically be transplanted to, say, islands in the West Pacific. They can be transplanted to, say, smaller countries or bigger countries. So places like Taiwan that have a very rich tick fauna, but it's not very well understood. Or Vietnam, Cambodia, Indonesia. The last time Indonesia had any tick work was sort of in the 1950s. So many countries are really, really ripe for this kind of research. So, as I said, ticks are chronically understudied within this region and particularly chronically understudied in Singapore. So it makes it a perfect case study in how we should start addressing this. So there's lots of different diagnostic methods we can use. We can look at, say, morphology, which is very accurate, but very time consuming and very intensive on human capital, human labor to be doing it. You need a lot of education to do this kind of thing, a lot of specialized training. We can use DNA barcoding. which can be upscaled massively, very, very, very cheaply. And it's easy to do. You can teach someone to barcode in an afternoon. So that's fantastic. Or we can start to integrate machine vision. Now we have the ability to feed in massive data sets into very, very highly accurate deep convolutional neural networks, which can basically automate tick identification in like a second. So it's really quick. But of course, if we use all of these together, we get this gold standard of cross-validating our data with multiple different independent methods. So let's think about barcoding to start with and the molecular approach to identifying ticks. If we have a look at, say, a big portal like the Barcode of Life, which is basically, its intention is to barcode all of the species on Earth so that we have these diagnostic barcodes that are species specific. Of those, they've done 63 species of hard ticks. So they've done less than 10%. So they're not doing very well considering they've been going for almost 20 years now. Pretty bad. So we decided to do it here. And we basically finished it. So we took the CO1 barcode, which we know is general enough that it varies between species, but specific enough that it's a reasonably good barcoding locus. And so this can facilitate, what we did was basically develop a national molecular library of tick barcodes. And of course, using this method which we were doing, which I'll run you through in a second, it's very fast, it's very cheap, and best of all, it's non-destructive. In many methods, people are destroying, they're mashing up their ticks, they're turning them into tick soup. And so of course, if they got some interesting barcode that's completely new, they can't go back and check the specimen to work out what it actually is. They can't put a face to a name. It just becomes this naked sequence, this naked read, and it goes into the dark matter of GenBank of all these species that apparently seem different based on their barcodes, but we can't actually validate it and check what they are. So we basically started a national tick surveillance network. So we had collaboration from clinicians who were giving us ticks from their human patients. We integrated organizations like SPCAs so that we had, and vets, so that we had companion animal ticks coming in. We went and surveyed wildlife so that we could look at what's actually in our sort of sylvatic tick cycles. And then of course we also did environmental sampling. And so we gathered ticks from 40 different host species from hundreds and hundreds of different individual hosts. So those are humans and animals. And in the end, we ended up with over 2,000 samples collected from the time that our program began in sort of 2018. So the first thing that we were doing was hotshot. So this is a non-destructive method, so it means that you still have your tick specimen intact at the end of it, and it's very quick, and it's very cheap, and it's very easy. So essentially, we take our tick out of ethanol, we let it dry for 2 minutes, we shove it in a tube. You can do this in, say, 96 well plates, so you can do this at mass scale if you're doing big surveillance efforts. You stick it in 10 microliters of an alkaline lysis buffer, we chuck it in the thermocycler for 30 minutes at 95 degrees, comes out, and then we add 10 microliters of an acidic neutralizing buffer. And voila, we have DNA. So let's compare it to some alternatives. You could use the QIAGEN blood and tissue kit, which is very slow, it's very cumbersome, and as you can see, it's actually quite expensive. $3.50 a sample. If you're doing, say, 1 or 2 samples, that's maybe okay. But when you start doing large surveillance efforts where you're having, say, tens of thousands of ticks coming in, suddenly that cost is astronomical. Or you could use the hotshot kit, and you can do a sample for about 2 cents each. We buy our sample. This is just one example of the hotshot solution that you can buy pre-made up for you. But we buy it even cheaper. We buy it as a powder. We build it. We make it up ourselves. And so it costs like 1 cent, half a cent, a third of a cent to do a sample. So you can do a whole 96 well plate of DNA extraction for under $1. Fantastic value. Really, really good value. And it's not cumbersome. And it doesn't take 3 or 4 hours like the QIAGEN kit. So great, if we want to start upscaling this, we can do this. And obviously, the more ticks we're barcoding, the more likely we are to reach an asymptote on our rarefaction curves and have sampled all the tick species in Singapore. We also are using nanopore sequencing technology. So these are things like the MinION from Oxford Nanopore company. And for this, we were using the CO1 locus, as I said, but we were using a smaller barcode region. So this is a 313 base pair. Normally the barcode size that you're using is typically about 650 base pairs. But of course, as you scale up the size of a barcode, your chance of not amplifying it, because maybe the DNA is degraded or something, increases. So the smaller your barcode, the more likely you have amplification success. And still at this size, we have the, say, diagnostic resolution to actually determine what our tick species are from barcodes. So MinION is increasingly releasing, or Oxford Nanopore is increasingly releasing these MinION sequences, and they're getting better and better and better every single year. So depending on what sequencing depth you want to do, you can sample, you can get, say, 10 million reads for like $500. That's great. And if you don't want super, super deep sampling, that means a lot of barcodes, right? So we can kind of upscale this. We have the DNA extraction pipeline that can do it incredibly cheaply, and we have the sequencing pipeline that can do it incredibly cheaply. So now we're at the kind of time when we can be doing things at scale. So you end up with all these barcodes. What do you do next? Well, we can sort them into clusters based on what they are, and then we can actually compare them, because it's a non-destructive technique, to the faces. So we can put faces to names, and we can start to say, okay, here's species, well, I mean, here they are. Here are the ticks of Singapore. They're all done. We did them. It didn't take long. It was really easy. It's really cheap. We can upscale this. You can do this in Papua New Guinea. You can do this in Taiwan. You can do this in Vietnam, whatever. Easy. Here's all the names. Here's all the species. And so you can do this kind of integrated approach. So in Singapore, this is our tick fauna. We have one species of Argus, one species of Ornithodorus. We have 6 Haemaphysalis, 6 Amblyoma, 1 Rhipicephalus, 1 Dermacenta, and 1 Izodes. And then, of course, we can run some ecological statistics on this. So these are rarefaction curves, which basically predict how many species you're likely to have based on the structure of your sampling. So we've basically got an asymptote for our tick species. So at present, it seems as though, based on our extensive sampling of upwards of 2,000 ticks, we have 17 species of ticks in Singapore, it seems. However, our interactions are still lagging. So we recorded like 86 or something interactions. But of course, when we plot that on the asymptote, on this rarefaction curve, we could still be going up. So we have somewhere maybe 120 odd interactions. So we still have some interactions between ticks and hosts that we don't know about, which could be pertinent to studying disease spillover. So we can barcode it. Super easy, it's done. We have a national tick barcode library now. The next thing we can start to do is machine vision. And increasingly, this has already been used for a long time in pathology diagnostics. So you feed in images of lungs, or you feed in images of tissue, and the machine vision essentially will be used to identify melanomas and things. Easy. So basically, we were using these deep convolutional neural networks, which are now at a scale where we can start to get really good accuracy, or pretty good accuracy. And we were using the Keras API, TensorFlow, and then we either use this microscope camera, or you can just use an iPhone. Easy. So these are the kind of images we're getting from our microscope camera. $400, pretty cheap. Here's the kind of images we're getting from an iPhone. Not bad. I mean, it's not the latest iPhone, but they're not terrible images. So essentially, we use this, what is considered quite an old model. So this is the VGG16 model, which was used in the ImageNet Large-Scale Visual Recognition Challenge in 2014, and it won. And essentially, during that challenge, it was fed over 1 million images to train it on, and then it had 14 million images to use to try and classify into 1,000 different categories. So these can be things like a cup of milk, or a dog, or a signpost kind of stuff. So essentially what we did is, we didn't train any of the pooling or convolutional layers. We left them as they were. We let them stay as they'd been trained using those million images. The only thing we did is we trained the final layer, the output layer. So we basically said, okay, you've got all these different these convolutional layers that can identify all sorts of different shapes and sizes and things. Classify based on that. We'll feed you some images, and you can classify them into 10 categories for 10 different tick species, that kind of thing. And so we did it. And so here's what we got. So using this model, which we basically only trained the final layer in, we could get 91% accuracy to genus identification based on microscope images, or 75% based on iPhone images. We got 87% species level identification accuracy, and 79% for iPhone images. We got 96% accuracy when it was identifying the sex of ticks, 80% when it was using iPhone images, and finally we got 100% accuracy in determining adults versus immature ticks, or 98% using iPhone images. So pretty good. Let's compare that against actual people's ability to identify ticks. This is taken from a study from this year, where they basically assessed the ability of non-tick experts to identify ticks in the United States. These 4 ticks, which I consider, maybe I'm a tick biologist, but I consider these very, very distinctive ticks. So the fact that people can't identify them is shocking. But the majority of people got the identifications wrong. People are really bad, which means that even with crappy iPhone images and even with this kind of 2014 machine vision model, we're still doing better than non-trained people, which is great. And then the final thing we did is we've been building this diagnostic manual for the ticks of Singapore. So you can feed machine vision in, you can do DNA barcoding, and if you really want to cross-validate it with a third source of information, you can use our tick manual, which is sort of forthcoming. It should be out sort of midway through next year. So Singapore is fully set up. We'll have the most advanced tick diagnostic capacity of any country in Asia. And all it takes is a tiny team and 4 years of looking at ticks. Easy. We can even compare some of the methods. So if we want to look at accuracy, morphology is the gold standard because all the ticks were described on morphology. We know what they look like. We know what they should be. Easy. AI drops a little bit. You were seeing for our iPhone-based images, we were getting, say, 87% accuracy for species-level identification. That's pretty good. It's better than average people, but it's not super good. So we can go through these different things. There's different pros and cons. Some have better costs. Some are easy to upscale, et cetera, et cetera. Let's move on because I don't have much time. So let's talk about forecasting vector-borne diseases. This somewhat departs from, what would you say, strict diagnostic microbiology and goes into more theoretical ecology and theoretical epidemiology kind of stuff. So when we're thinking about the spillover of pathogens, they actually have to pass a huge number of different filters before they can jump into people. Think of it as a pathogen has to get through a block of Swiss cheese to get into humans. And the little holes in the Swiss cheese don't always align, but when they do, it shoots right through and you get something like COVID-19. But that doesn't happen very often because there's a lot of pathogens going through and there's a lot of different filters within your ecosystem. We normally see pathogens as sort of microbiologists, clinicians, whatever. We see them at the very end. We see them when they get into humans. But there's all these other things floating around through this block of Swiss cheese that we haven't seen yet. So we want to try and work out, based on the structure of the Swiss cheese, we want to work out how the pathogens can get into humans and forecast and predict where they're going to come from. So they're always cycling in vector populations. Once we have interaction data sets of vectors, hosts, and humans, we can start to predict this kind of stuff. So where do we start? We need a training set. We need to validate this. So if we want to cure tropical zoonoses, let's look to cold, temperate, rainy, wet, miserable Britain. Because Britain has a reasonably good fauna of fleas. Fleas are really easy to look at their host associations because we actively pull them off animals. They live on their host most of the time, unlike mosquitoes. If you want to do mosquitoes, you can do things like metabarcoding. Host metabarcoding, you can barcode the blood meals in the mosquitoes to draw these relationship networks. But for fleas, it's easy. You just pull them off. You don't need any other fuss. And of course, there's been sort of 4 titans of British flea biology, Charles Rothschild, Carl Jordan, George Hopkins, and the late, great Miriam Rothschild, who spent a lot of time sorting the British flea fauna. So we know that they're very taxonomically stable, and we know a lot about their ecology, because every county has its own little flea expert that likes to pick fleas off badgers or hedgehogs or whatever. So we've got a really big data set, and we've got 150 years worth of data, because the British have been doing entomology longer than anyone else. So we basically took 150 years of flea data and plotted it. There it is. That is the physical representation of all the fleas and all the hosts from 150 years worth of collecting. Doesn't look like much, does it? It's kind of convoluted, it's kind of complicated. But basically what we have is 56 flea species, 175 host species, 3,500 interaction records, and 621 unique interaction records. So those are between one species and another species that are unique. So what can we do? First, we can start with things like module identification. And this is basically stealing some mathematics from graph theory. And essentially what it does is it identifies groups of nodes which interact more amongst themselves than other nodes. So if we think of this as a transmission network, we can think of these are fleas and hosts these are a group of hosts in which the fleas are bouncing around amongst these hosts more among themselves than among any other species within your ecosystem. So we can start to do that, and we can say, well, if we work out where humans are, which module they're in, then we can look at all the fleas in that module, because those fleas are the ones that are most likely to jump onto the humans and transmit pathogens. We can also screen the animals in that node. In the case of when we generated this, we found that mostly they're synanthropic animals. So these are things like hedgehogs that live amongst people's homes, foxes that are going through people's garbage, cats and dogs that live in our homes. So these are the species we can start to sequence to actually hunt new zoonoses. We can then also look at things like host specificity. How likely is a flea to jump onto another host and bite it? We can look at things like betweenness centrality. This in graph theory is this idea of what nodes or species within your network are acting as hubs. These are species that are connecting and interacting with many different host species. And we'd expect these species right in the middle of these huge networks with pathogens bouncing through them, these species right in the center to be the ones that mediate a lot of pathogens through these networks. So we can look at those. We can also look at weighted degree of separation. Within a network, how many degrees of separation do you have from other other host species? Because we'd expect species with closer degrees of separation, smaller degrees of separation, to share more pathogens. And finally, we can use this, a really simple equation we came up with to basically predict paths. And the idea is that we can produce some kind of, what would you say, a quantified risk of transmission. What are the most likely fleas to transmit pathogens to humans, and what are the most likely hosts that they're supposed to come from? And so we basically did that. Then we wanted to test it out. So we picked 7 different taxa, flea-borne diseases. So we picked undescribed rickettsia species, Rickettsia typhi, Rickettsia felis, Yersinia pestis, which is bubonic plague, Bartonella hensleyi, Bartonella quintana, and undescribed Bartonella species. And then we used 5 different metrics that we generated using our R package, which we built for this. And we basically tried to predict, based on our network of which fleas are associated with which pathogens, or sort of parazoonotic pathogens if they're not, if we don't know that they're zoonotic for sure. And so then we tried to see how that came out. And we basically got 75% accuracy in predicting, using our models, using our metrics, to actually predict who's a zoonotic vector. When we threw in publication bias, because of course there's strong publication bias, lots of people are working on Xenosyla chiopus, the plague flea. Lots of people are working on Tennocephalides felis, the cat flea, because they're predominant vectors. So when we threw that in, we found that publication bias also has an impact. But even without publication bias, we still get about 75% accuracy. That's fantastic. So we've been doing the same for our ticks in Singapore. We've built up a big reference collection of physical tick specimens and a big database of tick interaction records, which is almost as large as 150, it's got, yeah, 200, over 2,000, almost 3,000 interaction records, which we're getting to the point in 4 years of collecting that the British are getting to for fleas in 150 years of collecting. So we're doing pretty well, I'd say. And so, of course, this kind of data is really useful. We can start to do geospatial modeling. So we can start to predict where ticks are going to spill over, the physical geographical place in Singapore where they're going to spill over. We can look at seasonality and phenology. Are there more ticks after the second monsoon of the year? Questions like that. And then, of course, with our reference collection, we can do retrospective pathogen screening. We can develop novel diagnostics. So if we want to do like this kind of multi-TOF spectroscopy, we can be doing it because we've got all the tick specimens. Easy. We can build a multi-TOF spectroscopy library for ticks in Singapore in like 2 weeks or something because we've got the specimens. All we need to do is work out what their diagnostic output for this multi-TOF should be. So when I first got here in 2018, this is what our network of ticks and hosts looked like in Singapore. We didn't know very much about them. For reference, the pink nodes are the ticks, and the green nodes are the hosts. So we didn't know very much about who was eating who might spill over, and who's been eating people. Since then, this is what our network looks like now with almost 3,000 interaction records from like 40-odd host species and 17 tick species. And so here's humans right in the middle. We know what host species are feeding on humans. So we can start to screen those first, and we can start to use our sort of pipeline to work out who are going to be the most dangerous vectors and which pathogens are the most likely to become future zoonoses. And that's where we stop. We're pathogen screening at the moment, so we're hoping to sort of have stuff to publish within the next sort of, at least submit within the next few months, sort of couple months. So stay tuned. I'd just quickly like to acknowledge some of our many lovely partners that helped us with this: National Parks Board, NCID, Duke NUS, Monash University, Mundai, SPCA. And of course, if you want to follow me, we're in the age of social media, we're in the age of internet connectivity. If you do want to follow me, find me on Twitter, ResearchGate. Most of my papers are up there. They're all free, so please access them. Instagram, Facebook, email, whatever. Get in touch. Let's talk ticks. Let's talk zoonoses. Thank you very much. Done. Okay.
Thank you so much. That was a great animated talk. And I'm so glad you didn't ask me to identify the ticks. I used to know, though. I did study them for my ID boards.
Oh,
So I
good.
knew
It's
how the
probably still in there.
how the Lone Star tick looked, etc., etc.,
Yeah,
but
yeah.
not anymore. Anyway, so that was great. The thing for us clinically is that most of the time, nobody brings a tick.
Yeah.
Yeah, so it's already fallen off and forgotten.
Mhm.
And when we try to treat or presumptively diagnose rickettsial, so it's based on the constellation of symptoms and of course the magical response to doxycycline. So
Always.
yeah, so when like we were talking before, and in especially in this era of antimicrobial resistance, we are always worried about resistance, and there's not many things that can treat the rickettsia, right? So you were mentioning that they almost never become resistant to doxy. So would you want to just
Okay.
elaborate on that?
So, I mean, it's kind of like you can think of it from sort of the perspective of evolutionary biology, which is what the resistance is. We put a very strong selective pressure onto a pathogen, and then of course, eventually resistance develops if the evolutionary pressure is strong enough and long-lasting enough and extensive enough within the population. Of course, humans, for these vector-borne diseases, or certainly for tick-borne diseases, humans are a dead-end host. So the pathogen never cycles within human populations, and it never spills back into animal populations, which is where it exists as sylvatic populations of pathogen, vector, and host, and then occasionally spills over into humans. So when it arrives in us, it's doomed either way. Our immune system will either clear it, or a cluey clinician will clear it with doxycycline. So it's never getting out of us. So even if it develops resistance partway through the treatment protocol, almost never is a human going to get rebitten by a tick, and the tick is then going to leave and go and feed on some animals. And even if it did, nobody's treating animals with like doxycycline or anything, so there's no strong selection pressure. So the trait should be lost over time, or will be diluted by all the individuals that don't have doxycycline resistance. Yeah.
Right, that makes sense. We have one question from Dr. Gabriel. So the question is, in the sequencing of ticks, which you guys are doing, did you see any bacterial pathogens that could infect humans? I'm pretty sure you did. And if so, what proportion of these ticks had them?
So, good question. We haven't finished all of our microbiome work yet, and we haven't published it yet, but there are certainly rickettsia here. There are certainly Borrelia here. And we know that when we look around the region as well, people are finding Borrelias and rickettsias in neighboring Malaysia. We're finding lots in Vietnam and Thailand and the Philippines and Indonesia. So these things are definitely here. We have some of these pathogenic microbial genera. We also know, certainly in animals, there's Babesia. It's a canine Babesia, but the question is, do we have other Babesias in our wildlife, and can they jump over? So we don't know, because very often people come to clinicians, they present with this constellation of sort of looks like rickettsiosis, looks like some kind of spotted fever. Cluey clinician treats them with doxycycline and it's cleared from their system. In many cases, we don't have the bloods to be able to go and check so that we can link the species that we know that are clearly zoonotic, they're causing human diseases, to link them back to, say, a tick that gave it to them, because very rarely do they keep the tick, and then link that back to our big network in Singapore of hosts, ticks, spread through the environment. So drawing the causal link between which of these dozen flea-borne putative para-zoonotic pathogens actually infects people is very hard, unless people do the sequencing to try and link them together. But once we do, then we know, and you only need to do it once, and then we certainly know, oh, look, there's Rickettsia honii here, and it's in these rats, it's in these ticks, and we know it's a human zoonotic agent kind of thing. But yeah, certainly we have some of the nasties. Preliminarily, our preliminary data suggests that we have some of the nasty bacterial genera, which ideally we don't want, but they seem to be almost everywhere anyway, so of course we have them.
But no Lyme disease in Singapore.
I mean, we have Borrelia, which, I mean, Borrelia burgdorferi, kind of sensu stricto, the American one, we don't have that.
Okay.
But there are other relapse Borrelia species through this region. Just because it's not Bergdorferi, it would be shocking to find Borrelia burgdorferi sensu stricto here. That would be like finding a polar bear walking through Singapore. This is not where it evolved. We're an ocean away, we're a continent away. It would be very, very peculiar.
Right.
We're in Southeast Asia, we're in there's 2 different zoogeographic zones separating us, so it would never be here. But there can be things like it, and we certainly know some of the pathogens look as though they're probably not great. They're sisters to groups that we know are zoonotic.
Okay, great.
Yeah.
Thank you so much for
Thank
a
you very much
great
for having me.
talk. Yeah, so we'll stay in touch.